There is a creature in the ocean right now that science has quietly struggled to explain for decades. Something so small you could hold it on your fingertip, and yet what it does at the end of its life defies what we thought we understood about aging, death, and time itself. The jellyfish we are about to follow tonight does not simply live and die the way most animals do. Under certain conditions, it reverses. It goes backward through its own life cycle, becoming young again, and begins once more. Scientists have a name for this process, and we will arrive at it gently. But before we do, there are quieter things to notice first. The way this animal moves without a brain, feels without eyes, and builds a body with no bones and no blood. So let the weight of the day fall away now. Let the room grow soft around you, and drift with me into the slow-lit water where the world's most quietly impossible animal is already waiting, doing something most of us will never quite believe. The jellyfish is one of the oldest forms of complex animal life on Earth, and part of what has made it so enduring is how little it needs to exist. Its body is roughly 95% water. The remaining 5% is a loosely organized gel called mesoglia, threaded with simple proteins and a sparse scattering of specialized cells. There are no bones, no cartilage, no rigid structures of any kind. There is no blood, no heart to pump it, no lungs to exchange gas. What oxygen the jellyfish requires diffuses directly through its thin tissues from the surrounding water, and carbon dioxide diffuses back out the same way. The entire metabolic exchange that sustains a jellyfish happens passively, across surfaces so thin that the process requires no dedicated organ at all. This radical simplicity is not a design failure. It is an extraordinarily successful solution to the problem of living in a fluid, three-dimensional environment where building and maintaining complex structures costs energy that might otherwise go toward reproduction. The jellyfish body is energetically cheap to run. A small jellyfish in reasonably nutrient-rich water can survive on minimal food intake for extended periods because the machinery it must fuel is so minimal. There are no heavy bones to carry, no elaborate immune system to maintain, no large brain to keep supplied with glucose around the clock. The economy of the jellyfish body is part of what has allowed animals built on this plan to survive 5 mass extinction events and more than 500 million years of changing ocean conditions. The mesoglia, the gel that makes up the bulk of the jellyfish's body, has properties that are genuinely interesting to material scientists as well as biologists. It is a hydrogel, a network of protein and carbohydrate molecules that trap water within a loosely structured matrix. This gel can deform under pressure and return to its original shape, absorbing the mechanical energy of water movement without tearing. It provides buoyancy by matching the density of seawater closely enough that the jellyfish requires very little active effort to remain suspended at a particular depth. Some researchers have described the mesoglia as a kind of living jelly that simultaneously serves as skeleton, buoyancy device, and structural medium, doing the work of several tissue types in other animals with a single material. The cells of a jellyfish are organized into 2 primary tissue layers, an outer epidermis and an inner gastrodermis, separated by that central mesoglia. This 2-layered body plan is called diploblastic, and it represents one of the earliest organizational schemes in animal evolution. Most of the animals we are familiar with, including all vertebrates, are triploblastic, with 3 tissue layers from which the full range of organ systems develops. The jellyfish, with only two, cannot build a liver or a kidney or a brain. And yet within these two thin layers, remarkable specialization has occurred. The epidermis contains the stinging cells that make jellyfish both dangerous and effective hunters. The gastrodermis lines the internal cavity where digestion takes place. 2 layers, no blood, and 500 million years of unbroken success. One of the more surprising aspects of the jellyfish body is what happens to it when it is damaged. Cut a jellyfish in half, and the 2 pieces may each attempt to regenerate into a complete individual. Remove sections of tissue, and the surrounding cells reorganize to close the gap. Some jellyfish can regenerate substantial portions of their bodies following injury, a capacity that depends on a population of undifferentiated stem-like cells distributed throughout the mesoglea that can be recruited to rebuild what has been lost. This regenerative ability is not unlimited, and the extent varies between species, but it speaks to a fundamental plasticity in the jellyfish body plan, a looseness in the relationship between form and function that more highly organized animals have largely traded away in exchange for greater specialization. What the jellyfish body ultimately represents is a proof of concept that complexity is not the only path to persistence. For hundreds of millions of years, the most sophisticated animals on Earth have become progressively more elaborate, adding organs and systems and structures in response to the evolutionary pressures of an increasingly competitive biosphere. The jellyfish has done something different. It has remained radically simple and continued to thrive. In the deep time of evolutionary history, the jellyfish body plan is not a primitive relic awaiting improvement. It is a solution that has already been tested across timescales that make all of human history seem like a single afternoon, and it has not been found wanting. Jellyfish fossils are rare because soft-bodied animals without hard parts seldom survive the geological processes that turn living tissue into stone. But when they do fossilize, usually preserved in very fine-grained sediments that captured a brief impression before decay, they offer glimpses of a lineage so ancient that it stretches back to before most of the major animal groups had yet appeared on Earth. The oldest confirmed jellyfish fossils date to around 500 million years ago, from the Cambrian period, the era in which complex animal life first diversified extensively in the fossil record. But the group to which jellyfish belong, the cnidarians, is thought to be older still, with molecular clock estimates placing the origin of the lineage somewhere between 600 and 700 million years ago. This places the origin of jellyfish-like animals before the Cambrian explosion, in a world that was profoundly different from today. The oxygen content of the atmosphere and ocean was lower. The continents were arranged differently. The organisms sharing the ocean with the first jellyfish-like animals will be nearly unrecognizable to us. And yet, the basic body plan that emerged in that ancient sea, a radially symmetric gel-bodied, stinging tentacled predator of small prey, proved so well suited to ocean life that it persists essentially intact today. When you watch a jellyfish pulsing through the water in a modern aquarium, you are watching an animal whose fundamental architecture was road-tested in an ocean that no longer exists and found worth keeping. The fossil record for jellyfish, though sparse, has produced some remarkable specimens. In the Cambrian Burgess Shale of Canada and the equivalent Chengjiang deposits in China, soft-bodied preservation has captured early cnidarian forms with enough detail to reveal their basic body organization. In Jurassic deposits in Germany, impressions of jellyfish caught in anoxic sediments that preserved soft tissues have revealed animals strikingly similar to modern medusae, the familiar bell-shaped free-swimming form. Some researchers have even argued that trace fossils, patterns left in ancient sediment by the movement of soft bodies, may extend the evidence for cnidarian-like animals even further back into the Ediacaran period, more than 600 million years ago. The jellyfish is old in a way that few things in biology are. Part of what made the jellyfish body plan so durable is its relationship to environmental change. Over the hundreds of millions of years since the first cnidarians appeared, the ocean has experienced dramatic shifts in temperature, chemistry, oxygen content, and productivity. Many lineages that seemed successful in one era were eliminated as conditions changed. The jellyfish survived these transitions, apparently by combining a tolerant physiology with a reproductive flexibility that allowed populations to recover rapidly after local decline. Some species of jellyfish can tolerate a remarkably wide range of salinity, temperature, and oxygen levels compared to most marine animals. When conditions deteriorate, they may retreat to their polyp stage, a sessile form attached to hard substrate, and wait. When conditions improve, they reproduce and proliferate again. The evolutionary history of jellyfish also involves a fascinating structural innovation that has proven extraordinarily versatile: the nematocyst, the stinging cell that defines all cnidarians and has no equivalent in any other animal group. The nematocyst is a capsule within a specialized cell that contains a coiled thread armed with barbs and loaded with venom. When triggered by chemical or mechanical stimulation, this capsule fires with extraordinary speed, among the fastest cellular events in biology, completing discharge in less than a microsecond. The venom injected can immobilize prey many times the size of the jellyfish firing it. This single cellular invention, appearing somewhere in the Precambrian Ocean, gave the cnidarians a predatory capability that no soft-bodied animal had previously possessed, and it has been elaborated and refined in jellyfish lineages across hundreds of millions of years of evolution. The persistence of jellyfish across geological time should perhaps make us reconsider our casual assumption that complexity confers survival advantage. The history of life on Earth is littered with the fossils of highly complex, well-adapted animals that nevertheless vanished when conditions changed. The jellyfish, structurally simple by comparison, swam through the same catastrophes that eliminated the dinosaurs, the trilobites, and the vast majority of species that have ever existed, and arrived in the modern ocean essentially intact. There is a lesson in this, though it is a quiet one and does not lend itself easily to summarization. Sometimes the body that persists is not the most elaborate one. Sometimes it is the most accommodating. One of the most remarkable and least widely known facts about jellyfish is that most species lead two entirely distinct lives, so different from each other that early naturalists studying them in the field sometimes classified the two stages as completely separate species. The first life is the polyp, a small, stalked, sessile creature attached to a hard surface, like rock or shell, resembling a tiny sea anemone more than anything we would recognize as a jellyfish. The second life is the medusa, the familiar free-swimming, bell-shaped, tentacled form that most people picture when they hear the word jellyfish. These two forms are both part of the same animal's life history, connected by transformation, and the way they transition between stages carries some of the most biologically interesting details in all of invertebrate science. The polyp stage of a jellyfish is often described as the overlooked phase of the life cycle because it is small, inconspicuous, and fixed in place. But the polyp is anything but passive. It feeds actively, capturing small prey with its tentacles, and can reproduce asexually by budding, producing identical copies of itself that settle nearby and establish new polyp colonies. A single polyp can persist for months or even years, waiting for conditions that favor the production of medusae. When those conditions arrive, triggered by temperature shifts, changes in food availability, or seasonal cues that vary between species, the polyp undergoes a process called strobulation. It segments its body into a stack of disc-like forms, each of which is released as a tiny, free-swimming juvenile medusa called an ephora. The ephira that emerges from strobulation looks nothing like the adult medusa it will become. It is star-shaped, with 8 arm-like lobes surrounding a small central disc. It is typically less than 1 millimeter across. It swims weakly, feeds on microscopic prey, and begins the growth process that will eventually produce a fully formed adult medusa. This growth requires not just an increase in size, but a complete reorganization of body architecture. The lobes of the ephira expand and fuse into a continuous bell margin. The tentacles elongate and develop full stinging capability. The internal canal system elaborates to distribute nutrients efficiently across the growing body. The transformation from ephira to adult medusa is, in a quiet sense, a second development process following the first one that produced the polyp from the fertilized egg. The sexual reproduction of the medusa stage completes the circle. Adult medusae release eggs and sperm into the water, where fertilization produces a free-swimming larva called a planula. The planula swims for a period that varies from hours to weeks, depending on the species, then settles on a suitable hard surface and transforms into a polyp, starting the cycle again. This alternation between a sessile asexual polyp stage and a free-swimming sexual medusa stage is called metagenesis, and it is found in various forms across the Cnidarian group. The ecological significance of this dual life strategy is considerable. The polyp stage allows for local persistence and slow colonization of suitable habitat, while the medusa stage allows for genetic mixing through sexual reproduction and dispersal of larvae to new areas. The 2 stages of the jellyfish life cycle also serve different ecological roles in their communities. The polyp, attached to the substrate, feeds on small particles and zooplankton in the water column immediately above it, contributing to the turnover of nutrients at the seafloor water interface. The medusa, moving through the open water column, feeds on larger prey, including fish eggs, small crustaceans, and other zooplankton, and is in turn consumed by a range of predators, including sea turtles, sunfish, and various seabirds. In this way, a single species participates in the food web at 2 distinct trophic levels in 2 distinct habitat zones, an ecological versatility that few animals achieve through a single body form. There is an aspect of the polyp stage that has taken on new significance in the context of the species we will spend time with later in this story. The polyp is not simply a transitional stage between larva and medusa. It is a persistent, potentially long-lived form in its own right, capable of surviving conditions that would kill a free-swimming medusa, and of waiting for favorable circumstances with a patience that has no equivalent in the medusa stage. Some polyps have been maintained in laboratory conditions for years. Their maximum natural lifespan is unknown, and their relationship to aging and to the possibility of reversing it is one of the threads that leads us, quietly and gradually, toward the most extraordinary chapter in the jellyfish story. Every jellyfish is armed, and yet none of them has anything we would recognize as a mind. The stinging cells that make jellyfish simultaneously fascinating and dangerous operate not under central nervous control, but through a distributed sensory system that represents one of the earliest and most successful nervous architectures in animal evolution. Understanding how a jellyfish stings and how it coordinates the pulsing motion that drives its movement through the water requires setting aside our habitual way of thinking about behavior as something generated by a brain, and considering instead what happens when sensation and response are distributed evenly throughout an entire body. The jellyfish nervous system is called a nerve net. It has no central ganglion, no brain, no spinal cord, no hierarchical structure of any kind. Instead, neurons are distributed throughout the tissues of the animal in a loose mesh, connected to one another and to sensory cells and effector cells in a relatively non-directional way. When a sensory cell at the tip of a tentacle detects the chemical or mechanical signal that indicates potential prey, it triggers the discharge of nearby nematocysts and sends a signal spreading outward through the nerve net. Depending on the strength and spread of this signal, it can trigger a local response, just the tentacles in one area, or a more widespread response involving the entire bell and all the tentacles simultaneously. The nematocysts themselves are marvels of cellular engineering. Each one is produced by a cell called a cnidocyte, which manufactures the entire capsule, venom, and firing mechanism as a single intracellular structure. The capsule is under enormous internal osmotic pressure, ready to discharge, and is held in its cocked state by a molecular trigger that responds to two separate stimuli: mechanical contact and specific chemical signals from prey. This dual trigger system reduces accidental firing and ensures that the stinging response is calibrated to actual prey contact rather than random disturbance. Once fired, the thread penetrates the prey's tissue and delivers venom that varies between species from mildly irritating to potently toxic. The entire discharge event, from trigger to penetration, takes less time than any process in neuroscience has been able to measure at the scale of a single nerve impulse. Despite having no brain, jellyfish are capable of behaviors that look, from the outside, surprisingly directed. They pulse rhythmically to move through the water, creating a propulsive jet that carries them upward, while passive sinking between pulses allows them to cover vertical and horizontal distance with minimal energy expenditure. They can orient toward light, avoid shadows that might indicate predators, and in some species navigate towards specific depth zones where prey are concentrated. These behaviors emerge not from centralized computation, but from the integrated activity of simple sensory cells responding to gradients of light, pressure, and chemistry, connected to effector cells through the nerve net in ways that produce consistent behavioral outcomes without requiring any central director. Have evolved structures that go beyond the basic nerve net in organizational sophistication. The box jellyfish, or cubozoans, are perhaps the most striking example. These animals have evolved true eyes, complete with corneas, lenses, and retinas, arranged in clusters on their four rhopalia, specialized sensory structures around the bell margin. These eyes are strikingly similar in basic structure to vertebrate eyes, a remarkable case of convergent evolution in which natural selection independently produced similar optical solutions in two lineages separated by hundreds of millions of years of divergence. Box jellyfish use these eyes for visually guided behaviors, including avoiding obstacles, orienting to specific light environments, and in some species, apparently navigating using visual landmarks. They have done all of this without evolving a central brain to process the visual information. The question of what jellyfish experience, if they experience anything at all in any meaningful sense, is one that biology is not yet equipped to answer with confidence. Consciousness, as we understand it, appears to require a level of neural integration and centralization that the jellyfish nerve net does not obviously provide. And yet the stinging response, the pulsing rhythm, the orientation behavior, and in some species the visually guided navigation all suggest a kind of responsiveness to the world that goes beyond simple reflex. The jellyfish does not think in any way we currently understand thinking, but it is not simply a passive object in the current either. Somewhere between thinking and not thinking, the jellyfish inhabits a behavioral space that our language barely has words for, present, responsive, and entirely its own kind of alive. The pulsing motion of a jellyfish is one of the most hypnotic things the ocean offers. Watch a jellyfish from above, and the bell contracts and relaxes in a slow, even rhythm, drawing the animal gently upward with each contraction and allowing it to drift between pulses. The motion is not frantic or desperate. It is measured, almost meditative, as though the animal is keeping time with something internal and unhurried. This pulsing is controlled by a ring of nervous tissue at the bell margin called the marginal ganglion, which generates spontaneous rhythmic electrical signals that spread across the bell muscle and trigger coordinated contraction. In a healthy jellyfish, this rhythm is self-sustaining and remarkably consistent, continuing without interruption for as long as the animal is alive. The fluid dynamics of jellyfish locomotion have attracted considerable interest from physicists and engineers in recent decades. The pulsing bell does not simply push water backward in a simple jet. It creates a complex flow pattern involving two interacting vortex rings, one expelled downward behind the animal and one drawn up in front of it, that together produce a pulling force more efficient than a simple push jet alone. This mechanism, described by researchers studying jellyfish swimming with high-speed imaging and flow visualization techniques, turns out to be significantly more energy efficient than the equivalent motion in many mechanical devices designed by engineers. Some researchers have drawn on jellyfish locomotion as inspiration for designing soft robotic systems and underwater vehicles, attempting to replicate a hydrodynamic solution that evolution found hundreds of millions of years ago. The pulsing rate of a jellyfish is not fixed. It varies with temperature, with the animal's nutritional state, and with environmental conditions in ways that reflect a continuous adjustment of metabolic expenditure. In warmer water, the pulse rate tends to increase as chemical reactions in the muscle tissue proceed faster. In cold water, it slows, sometimes dramatically. A jellyfish in very cold water may pulse so slowly that it appears almost still. This temperature sensitivity means that jellyfish in different ocean environments are effectively running at different metabolic speeds, and the energetic cost of a given life history strategy varies significantly between warm and cold water populations. Some researchers have used these metabolic differences to understand how jellyfish distributions may shift as ocean temperatures change over coming decades. The bell contraction itself is generated by a layer of smooth-like muscle cells on the underside of the bell. These cells are not skeletal muscle in the way we usually think of it, with distinct fibers and dedicated nerve endings at each fiber, but a more diffuse sheet of contractile cells embedded in the mesoglia that respond to the electrical waves spreading across them from the marginal ganglion. The contraction sweeps across the bell in a wave, rather than occurring simultaneously across the entire surface, which produces the characteristic cupping motion that generates the efficient vortex dynamics described above. The coordination of this wave, without a brain to direct it, emerges from the geometry of the tissue and the speed at which the electrical signal propagates through it. Another example of the jellyfish generating complex behavior from simple physical principles. There is a relationship between the jellyfish's pulsing and the physical structure of the water it inhabits that researchers have been exploring with increasing precision. Jellyfish are not fully at the mercy of ocean currents, but neither are they fully independent of them. They can swim against weak currents and use active pulsing to adjust their depth and position within the water column. But in stronger currents, they are largely carried by the flow. This partial autonomy has led some researchers to describe jellyfish as diel vertical migrants, animals that use active swimming to move upward during certain parts of the day and allow themselves to sink during others, exploiting the vertical structure of the ocean to access different food concentrations at different times. Watching a jellyfish pulse in a quiet aquarium, it is easy to lose track of time. The rhythm is even, the motion gentle, the animal apparently indifferent to the glass and the light, and the observer pressed close to the surface. In the open ocean, this same rhythm continues regardless of whether anything is watching, day and night, across weeks and months that add up to a life whose duration varies enormously between species. The pulse is not a performance. It is simply the jellyfish being alive, doing the one active thing its body does continuously from the moment it develops a bell to the moment it stops. In that rhythm, there is something that feels very old, something that was pulsing in the ocean long before there was anyone to notice it. In 1988, a young marine biology student named Christian Sommer was collecting samples in the waters off Portofino, Italy, studying the life cycles of small hydrozoans, the group of cnidarians to which the most famous immortal jellyfish belongs. Among his samples was a small species called Turritopsis dornii, a transparent, thimble-sized jellyfish barely a few millimeters across. In the normal course of studying these animals in the laboratory, Sommer noticed something that did not fit the expected developmental sequence. Rather than dying at the end of its medusa phase, as jellyfish are supposed to do, some of the Turritopsis specimens were reverting. They were sinking to the bottom of the container, reabsorbing their tentacles, and transforming back into the polyp form from which they had come. The initial observations were noted, but not immediately published, partly because the phenomenon seemed so anomalous that confident description required careful verification. It was not until the 1990s, through the work of researchers including Stefano Perino and his colleagues, that the reversal process in Turritopsis dornii was documented and published in a scientific paper. The paper described what the researchers called reversion to the juvenile stage, the transformation of a sexually mature medusa back into a polyp under conditions of starvation, physical damage, or age-related deterioration. The process was repeatable and consistent, occurring across multiple individuals in controlled conditions, and the resulting polyps were capable of beginning the life cycle again. The cellular mechanism underlying this reversion involves a process called transdifferentiation, in which cells of one specialized type convert directly into cells of a different type without passing through an undifferentiated stem cell intermediate. In the reverting Turritopsis medusa, the cells of the bell muscle, the outer body wall, and the internal tissues appear to despecialize and then respecialize into the cell types needed to build a polyp. The entire body plan reorganizes from one form to another, dismantling the medusa architecture and rebuilding the polyp architecture using the same cellular material. This is not regeneration, in which lost tissue is replaced by regrowth. It is something more fundamental, the reversal of developmental history within cells that had already reached their specialized adult state. Transdifferentiation is known in some other biological contexts, primarily in experimental laboratory settings where researchers have chemically induced one cell type to convert to another. But the natural, whole organism transdifferentiation that Turritopsis undergoes spontaneously under stress is unusual enough that it attracted significant scientific attention once described. The question researchers began to ask immediately was whether this process represents true biological immortality, the ability of an organism to escape death through developmental reversal, rather than simply achieving a very long lifespan. The answer requires careful thinking about what we mean by immortality, because the process is more nuanced than the popular description sometimes suggests. What Turritopsis dohrnii appears to be capable of is not immortality in the sense of being unable to die. The reverted polyp can still be eaten by a predator, killed by disease, or damaged beyond recovery. The medusa stage itself can die from causes other than age. What the reversal process circumvents, specifically, is the normal programmed deterioration and death that follows sexual maturity in most animals. The biological clock that ticks down from the moment an animal reaches reproductive age toward a fixed endpoint. In Turritopsis, that clock appears to be resettable. The medusa that has reached the end of its normal lifespan can, under certain conditions, turn back the clock to zero and begin again. Whether any individual does this repeatedly in nature, accumulating what would amount to an indefinite lifespan, is a question that field ecology has not yet been able to directly answer. The discovery of Turritopsis dohrnii's reversal capacity did not produce an immediate wave of scientific excitement. The initial papers attracted attention in the scientific community, but widespread popular awareness came later, with a long-form magazine article in 2012 that introduced the animal to a much larger audience under the phrase, "the immortal jellyfish." By that point, the biological reality was well established in the literature, though the popular framing inevitably simplified the nuances. What makes the story scientifically compelling is not the simplification, but the genuine strangeness of what Turritopsis actually does. It provides, in the natural world, a demonstration that the developmental trajectory which most biologists had assumed was strictly one-directional can, in at least one lineage, be run in reverse. The question of how Turritopsis dohrnii manages its reversal at a molecular level is one of the more active areas of research in the biology of aging, and the answers emerging from recent genomic and cell biology work are genuinely interesting. A key study published in 2022, led by Carlos López-Otín and colleagues in Spain, compared the genome of Turritopsis dohrnii to that of a closely related species that lacks the immortality trait, looking for genetic differences that might explain the reversal capacity. What they found were significant expansions in gene families associated with DNA repair, stem cell maintenance, and cellular reprogramming, the molecular machinery that allows cells to maintain genomic integrity over time and to adjust their developmental identity. DNA repair capacity is one of the factors most consistently associated with longevity across animals. Species that live longer tend to have more robust systems for detecting and correcting errors in their DNA replication. And the accumulation of DNA damage over time is one of the leading molecular theories of aging. In Turritopsis, the expanded complement of DNA repair genes may allow the cells undergoing transdifferentiation to correct any errors that have accumulated during the medusa's life before re-entering a fresh developmental trajectory. If the cell's genome can be effectively reset alongside its developmental identity, then the reverted polyp begins not only with a new body plan, but with a more youthful molecular state than the medusa it came from. The gene families involved in cellular reprogramming in Turritopsis include homologs of genes known in vertebrate biology to be associated with induced pluripotency, the laboratory technique that allows researchers to rewind adult cells into a stem cell-like state. The discovery that jellyfish have naturally evolved expanded versions of these same gene families was striking to researchers who work on regenerative medicine and aging, because it suggests that the molecular toolkit for cellular reversal is more ancient and more widely distributed across the animal kingdom than previously recognized. Turritopsis did not evolve an entirely novel mechanism for its reversal. It appears to have amplified and elaborated a capacity for cellular flexibility that exists in simpler form across a much broader range of animals. The trigger for reversal is also worth examining carefully. Turritopsis does not reverse spontaneously at the end of every normal medusa lifespan. The process appears to be induced by specific stressors: starvation, physical damage, age-related deterioration, or extreme environmental conditions. This suggests that the reversal is a conditional survival response rather than a scheduled part of the life cycle. When conditions are good, the medusa completes its normal life and dies. When conditions are bad, the organism essentially chooses an alternative exit from the medusa state, bypassing death by reverting to the polyp stage. Viewed this way, the immortality of Turritopsis is not absolute. It is conditional and contingent, a fallback strategy that engages under duress rather than a permanent exemption from the normal rules of biological time. What triggers the cellular machinery of reversal at the molecular level is still being investigated. Some researchers believe the key signal is a drop in metabolic resources, essentially a cellular perception of starvation that activates stress response pathways previously dormant in the healthy medusa. Others point to the role of specific signaling molecules that are known in other animals to regulate the transition between differentiated and stem-like cell states. The process of transdifferentiation, once it begins, appears to unfold according to a relatively consistent program, with characteristic stages observable under microscopy, as the medusa gradually loses its adult form and the polyp form takes shape. The speed of reversal varies with temperature, taking longer in cold water and proceeding more rapidly in warm. The broader significance of Turritopsis's molecular biology extends well beyond curiosity about a small Mediterranean jellyfish. The genes identified in the 2022 genomic study as expanded in Turritopsis relative to its non-reversing relatives are mostly genes with known roles in human aging and disease. The DNA repair pathways that appear amplified in Turritopsis are the same ones that, when compromised in humans, contribute to cancer and age-related genomic instability. The stem cell maintenance genes that appear enhanced are the same ones whose decline with age contributes to the loss of tissue regenerative capacity in aging humans. The jellyfish has found a way to maintain and deploy these systems at a level and in a manner that has no equivalent in vertebrate biology, and understanding how it does so is a question with implications that reach far beyond the animal itself. A Turritopsis dohrnii medusa in the wild lives at a pace almost impossibly different from our own. Its entire adult life, from the moment it is released as an ephora from its parent polyp to the point at which it either reproduces and dies in the normal manner or undergoes reversal, unfolds across weeks to months rather than years or decades. The individuals we can observe directly in laboratory tanks are small enough to rest comfortably on a fingernail. Their tentacles are transparent. Their bell is thimble-shaped and colorless, except for a distinctive reddish-orange stomach visible through the mesoglea. In the scale of the ocean, they are barely present. And yet the cellular processes occurring within them are among the most biologically remarkable things happening anywhere in the water column. Time, for an organism like this, is experienced in a biological currency entirely different from our own. The rhythms that organize a Turritopsis's existence are not circadian in the mammalian sense. There is no sleep-wake cycle, no daily hormonal tide, no seasonal reproductive calendar tied to day length. There is the rhythm of pulsing, continuous and regular, and the rhythm of food availability, and the rhythm of environmental conditions, and somewhere in the background, the slower tick of cellular aging that in normal circumstances leads eventually to senescence, and in abnormal circumstances leads to reversal. Whether Turritopsis experiences anything at all remains genuinely open, but it navigates a time structure radically unlike ours, measured in contractions and chemical concentrations rather than days and years. The population dynamics of Turritopsis in the wild are difficult to study precisely because the animal is small, transparent, and not easily distinguished from other hydrozoans without close examination. What researchers have pieced together suggests that populations can persist in a given location for extended periods, maintained by the polyp stage between outbreaks of medusae. The polyps themselves are inconspicuous, attached to hard surfaces in shallow water, and can survive for extended periods in the absence of conditions favorable to strobilation. In this sense, the Turritopsis population at any given location is like a banked fire. The active flame of medusa life may flicker out in poor conditions, but the embers of the polyp colony persist, ready to reignite when circumstances allow. One of the more thought-provoking implications of Turritopsis's biology is what it suggests about the evolutionary origin of aging itself. For most of the history of biology, aging has been viewed as an inevitable consequence of natural selection's declining power with age. Natural selection acts most strongly on traits that affect survival and reproduction early in life, and less strongly on traits that affect later life because fewer individuals survive to old age to express them. This asymmetry is thought to allow the accumulation of genetic changes that are harmful in old age but neutral or beneficial in youth. And over evolutionary time, these accumulations produce senescence. Turritopsis's reversal capacity suggests that under the right selective pressures, aging can be at least partially circumvented, that the logic of senescence is not absolute. The selective pressure that may have driven the evolution of reversal capacity in Turritopsis is itself a subject of debate. One hypothesis holds that in environments where food is unpredictable and mortality from starvation is a significant cause of death for medusae, The ability to revert to the polyp stage rather than dying when food runs out provides a strong fitness advantage. An organism that can retreat and wait is more likely to eventually reproduce than one that dies when conditions deteriorate. This hypothesis would predict that reversal capacity should be most pronounced in species or populations inhabiting particularly unpredictable environments. And there is some correlational evidence for this from comparative studies of related species. But the full evolutionary story remains a subject of ongoing research. There is something gently humbling about the fact that one of biology's most extraordinary solutions to the problem of aging exists in an animal that most of us have walked past in rock pools without noticing. The chemical and genetic sophistication embedded in Turritopsis's tiny transparent body took nature hundreds of millions of years to refine, and we have known about it scientifically for only a few decades. How many other organisms carry solutions to deep biological problems in their tissues, waiting for us to look carefully enough to notice? The jellyfish does not know it is remarkable. It simply pulses on through the water, doing what its cells have been built to do. Indifferent to the centuries of scientific tradition it has quietly upended. Jellyfish are not simply passive drifters in the ocean. They are active participants in the ecological dynamics of marine systems in ways that have taken decades of research to begin to fully appreciate. As predators, jellyfish consume fish eggs, larvae, zooplankton, and in the case of larger species, small fish, contributing significantly to the regulation of prey populations. As prey, they are consumed by sea turtles, sunfish, certain seabirds, and a number of fish species that have evolved tolerance for the stinging cells. And as decomposing organic material, they contribute to the export of carbon from surface waters to the seafloor in ways that researchers are now quantifying as part of the broader carbon cycle of the ocean. One of the more surprising ecological roles of jellyfish has emerged from studies of what lives on and within them. The bell and tentacles of larger jellyfish species are colonized by small fish, amphipods, and crustaceans that use the jellyfish as both shelter and food source. Picking parasites and small prey from the jellyfish's surface while benefiting from the protection its stinging cells provide against larger predators. Young jackfish and other juvenile pelagic species are frequently observed sheltering beneath the bells of large jellyfish, riding along in the current created by the bell's pulsing. This association appears to benefit the fish considerably, providing a mobile refuge in an otherwise exposed open ocean environment, while the cost to the jellyfish seems to be minor. The contribution of jellyfish to the ocean's biological carbon pump is an area of active current research. When a jellyfish dies, its body, being mostly water, decomposes extremely rapidly in the water column, releasing nutrients and dissolved organic carbon back into the surface layer in a matter of hours to days. But before decomposition is complete, some portion of the sinking organic matter reaches deeper waters, contributing to the export of carbon from the sunlit surface layer to the deep ocean. Additionally, jellyfish produce fecal pellets and mucus that can aggregate into particles that sink faster than dissolved material. Some researchers have proposed that in areas where jellyfish bloom abundantly, their contribution to the biological carbon pump may be more significant than previously assumed, though quantifying this in the field is methodologically challenging. Jellyfish blooms, the periodic explosions in population density that bring millions of animals together in dense aggregations, are one of the more dramatic ecological events in coastal marine systems. These blooms can clog the intake pipes of coastal power plants and desalination facilities, foul fishing nets, and reduce fish populations by consuming the same zooplankton prey. They occur naturally and have been recorded in historical accounts going back centuries. But there is accumulating evidence that blooms may be increasing in frequency and intensity in some regions, potentially in response to changes in ocean temperature, nutrient loading from agricultural runoff, and the removal of fish that compete with or consume jellyfish. Whether this represents a long-term trend or natural variability remains a subject of scientific debate. Jellyfish also participate in the ocean's nutrient economy through a mechanism that researchers have described as jelly falls. The sinking of dead jellyfish bodies to the seafloor in large numbers following the collapse of a bloom. These falls can deliver substantial pulses of organic material to seafloor communities that are otherwise dependent on the relatively slow rain of small particles from above. Studies of jelly falls in deep Norwegian fjords have found that benthic communities of worms, crustaceans, and bacteria respond rapidly to these organic pulses, consuming the jellyfish bodies within days or weeks. In this way, the bloom and collapse cycle of jellyfish populations connects the surface productivity of coastal waters to the communities living in the dark on the bottom, in a pulsed and irregular but ecologically meaningful way. The ecological complexity surrounding jellyfish has begun to shift how marine biologists think about these animals in the broader context of ocean food webs. For much of the 20th century, jellyfish were regarded as something like dead ends in the food web, consuming resources without contributing much of nutritional value to higher predators. More recent work has documented a broader range of jellyfish predators than previously recognized. A growing understanding of the diverse roles jellyfish play in nutrient cycling, and a new appreciation for the structural role that jellyfish-associated communities play in open ocean ecosystems. The jellyfish is not an ecological passenger. It is, in the waters where it thrives, a significant part of the architecture of life. Sleep, as we experience it, requires a brain. It involves the coordinated activity of billions of neurons cycling through characteristic patterns of electrical activity, the slow waves of deep sleep, the rapid eye movements of the dreaming phase, the transition between these states regulated by a complex interplay of neurochemicals and circadian timing systems. All of this requires a central nervous system of considerable sophistication. The jellyfish, with its distributed nerve net and complete absence of anything we would call a brain, cannot sleep in this sense. And yet, recent research has produced a surprising finding. Jellyfish may enter a state that functions as sleep, recognizable not by the neural signatures we associate with sleep in vertebrates, but by the behavioral signatures that define sleep across the animal kingdom. In 2017, a team of researchers led by Ravi Nath and Claire Bedbrook at Caltech reported a study on Cassiopeia, the upside-down jellyfish, a species that rests on the seafloor with its bell facing downward and its tentacles waving upward, unusual among jellyfish for its largely sedentary lifestyle. The researchers found that Cassiopeia shows a consistent nighttime reduction in pulsing activity. The animals pulse less frequently during certain hours than during others, and this reduced activity state meets 3 of the standard behavioral criteria used to define sleep across animal species: reduced activity compared to the waking state, reduced responsiveness to external stimuli, and homeostatic rebound, meaning that animals deprived of their rest period subsequently show increased activity as if compensating for the lost rest. The discovery was notable precisely because Cassiopeia lacks a centralized brain. If sleep serves functions that require a brain, such as memory consolidation or synaptic homeostasis, those functions cannot be what drives Cassiopeia's rest state. This suggests that whatever sleep is doing at its most fundamental level, at least some of its functions are accessible to a nervous system as simple as a distributed nerve net. Some researchers have proposed that even the simplest nervous systems may require periodic downtime to clear metabolic waste products, restore the chemical gradients needed for nerve cell function, or reduce the accumulated cost of continuous sensory processing. If true, sleep is not a sophistication added to biology by complex brains. It is something more basic, a need that the simplest neural tissue shares with the most elaborate. The rest state of jellyfish has also been investigated in the context of light responsiveness. Many jellyfish show behavioral changes in response to light and dark cycles that parallel circadian rhythms in other animals. Even though the molecular machinery of the circadian clock, as found in vertebrates and insects, appears to be absent or differently arranged in cnidarians. Cassiopeia placed in constant light does not show the same regular rest period it shows in normal light-dark cycling conditions, suggesting that the rest state is at least partly driven by external light cues rather than a fully autonomous internal clock. This light-dark entrainment of a behavioral rest cycle in an animal without a brain is one of the more quietly remarkable findings in recent sleep biology. For Turritopsis dohrnii specifically, sleep or rest behavior has not been studied in the depth that Cassiopeia has been examined, and the smaller, more active life history of the Medusa stage makes equivalent observations more technically challenging. But if the general pattern across cnidarians holds, some form of reduced activity state in response to light-dark cycling likely occurs in Turritopsis as well. What the rest state means for an animal that can reverse its own development is a genuinely interesting question. Does the cell reprogramming associated with reversal occur preferentially during rest periods, as some cellular maintenance processes do in other organisms? Does the metabolic state of a resting medusa influence the likelihood or trajectory of reversal? These questions sit at the intersection of sleep biology and aging research in a way that would have seemed entirely fanciful before the biology of Turritopsis became clear. There is something appropriate, in a quiet way, about ending this section where we are. An animal that can reverse time within its own biology, that can turn back toward its own beginning, that lives in a way that makes words like sleep and age take on new and uncertain meanings, is an animal that gently challenges the frameworks we use to organize our understanding of life. It doesn't ask us to abandon those frameworks. It asks us to hold them a little more lightly, to recognize that they were built to describe a particular range of biology, and that the ocean contains things that sit at the edge of that range and ask us without urgency to stretch a little further. The jellyfish pulses on, the water holds it, and somewhere in the reduced activity hours of a dark and quiet sea, something small and transparent rests. Turritopsis dohrnii is native to the Mediterranean Sea and the waters of the Northwest Atlantic, but it has been spreading. Over recent decades, genetic analysis of Turritopsis populations collected from ports and harbors around the world has revealed the presence of the Mediterranean species in locations far beyond its original range, including the coasts of Japan, Panama, Australia, Spain, and numerous other sites across both hemispheres. This global spread has not been the result of the jellyfish swimming across ocean basins. It has been entirely inadvertent, carried by the activity of international shipping, most likely in the ballast water that large vessels take on in one port and discharge in another to maintain stability during ocean crossings. Ballast water is one of the most significant vectors for the global spread of marine invasive species. Ships take on many thousands of tons of seawater in port, and this water carries whatever microscopic and small-bodied organisms are present in the local marine community. When the ship arrives at a distant port and discharges ballast water, those organisms are released into a new environment. Most do not survive the transition, but some do, particularly organisms like small jellyfish polyps attached to hard surfaces in the ballast tanks, or small medusae that can survive the voyage in a dormant or reduced state. Turritopsis, with its capacity to revert to the polyp stage under stressful conditions, may be particularly well equipped to survive the journey in ballast tanks and emerge viable at the destination port. The ecological consequences of Turritopsis's spread are not yet fully understood. As a small predator of plankton, it competes with larval fish and other zooplankton predators in its new environments, and its potential for population outbreaks, if the reversal capacity allows it to maintain population density in ways that non-reversing species cannot, raises questions about its competitive impact. So far, no dramatic ecological disruption attributable specifically to Turritopsis has been documented in its introduced range. But the organism is small, its populations are difficult to monitor, and the full ecological footprint of its global spread may not yet be visible against the background of other changes happening simultaneously in coastal marine ecosystems around the world. The genetic study of Turritopsis populations across its expanding global range has revealed something interesting about the species' population structure. Rather than finding a uniform global genotype spreading from a Mediterranean source, researchers have found evidence for some genetic differentiation between populations, suggesting that even in introduced locations, populations may be adapting to local conditions over relatively short timescales. This rapid local adaptation in an introduced species is not unusual, but it is notable in Turritopsis because it adds a layer of complexity to understanding the reversal trait itself. If the capacity for reversal varies in its frequency or triggering conditions between populations, the ecological dynamics of introduced Turritopsis in different ocean environments may differ in ways that single-species assessments would not predict. The global spread of Turritopsis has also generated scientific benefits, somewhat unintentionally. As researchers collected and genetically characterized samples from ports around the world, they assembled a detailed picture of the species' genetic diversity and global connectivity that would have been much harder to obtain by studying only Mediterranean populations. This opportunistic sampling has contributed to understanding the fine-scale population genetics of the species and, by extension, to understanding which genetic variants are associated with reversal capacity across natural populations. In this sense, the shipping industry's inadvertent dispersal of the jellyfish has provided the scientific community with a natural experiment that could not have been designed. There is a broader reflection available here, if we want it, about the relationship between a species whose most remarkable trait is the ability to reverse and begin again, and the process by which it has been spread around the world in the ballast tanks of ships moving through a globalized economy. The jellyfish does not choose its dispersal. It simply survives the conditions it finds itself in. reverts when those conditions are stressful, and continues when they improve. Whether this amounts to a kind of ecological resilience or simply the blind operation of cellular chemistry in an organism with no preferences about where it lives, is a question that depends on how generously we extend the language of agency to a transparent, brainless, millimeter-sized animal pulsing in the dark water of a ship's tank, heading somewhere it has never been, carrying its particular biological secret into a new sea. And here, in the deep and gentle quiet of this story, we can rest. The jellyfish continues to pulse somewhere beneath the surface, indifferent to our watching, indifferent to its own impossibility. It does not know what it has overturned in biology. It does not know what questions it has raised in laboratories from Spain to Japan, what papers it has inspired, what careful late-night conversations between scientists it has sparked about the nature of time and aging and the direction of a life. It simply moves through the water, and when the water becomes difficult, it grows quietly backward into something younger. The ocean holds far more than we have found names for. In its cold and dark and luminous depths, life has been finding solutions to the problems of existence for longer than we can easily imagine. And some of those solutions have no equivalent in anything that walks or flies or grows roots into the soil. Tonight, in whatever small way a story can offer this, you have been in the water with one of the most quietly extraordinary of those solutions. Not the largest, or the loudest, or the most dramatic, but perhaps the most gently, persistently strange. Let the water close softly overhead now. Let the pulsing continue without you needing to watch it. The jellyfish will still be there, in the dark and the salt, when you wake up, doing what it has always done, which is, in the end, simply the remarkable act of continuing.